All right, now that we've covered the structure of an enzyme, let's go a little bit more into detail about how these enzymes are used by the cells. So an enzyme is going to act on a substrate in order to make a product. Sometimes those enzymes are considered exoenzymes. So that means that the substrate is actually located outside of the cell, and the enzyme is produced within the cell and then pumped outside of the cell to work on something in the environment. It may break down something in the bacteria's environment that is potentially hazardous to it, or it might break down something that it can then use the products as nutrients. So sometimes those enzymes are pumped out. An endoenzyme is one that we more typically think about is performing a function within the cell. So the substrate is within the cell, and we are breaking down and producing products inside of the cell. Now, the enzymes can also be considered either a constitutive or a regulated enzyme. A constitutive enzyme is one that's always present in the same amounts inside of the cell, right? So as we add more substrate or more substrate is present, that the amount of enzyme that is there or available does not change. However, a regulated enzyme is one that could be either induced or repressed, it is said, or it could be upregulated or downregulated, that the response of the cell to the substrate will indicate how much enzyme is there. So if we add more substrate in, we have this upregulation or induction in the formation of that enzyme, and so we tend to have a higher amount of that enzyme inside of the cell. Whereas if the substrate is not present, right, then we downregulate, we break down those enzymes, we repress those enzymes because they're not needed at that time. So we have either constitutive or regulated enzymes. Specific to bacteria and pathogens, there are bacteria and other types of pathogens that will produce exoenzymes, right, that make them pathogenic. So anything that causes illness or sort of a disease state, or makes the bacteria or other organism, by that I mean some other kind of eukaryotic pathogen, more dangerous or likely to cause an infection, is called a virulence factor, right? So it's, potentially how hazardous something is. Exoenzymes can be virulence factors, right? So it's some type of enzyme that's pumped out of the cell and does some sort of damage or allows that bacteria to persist within the body or avoid some sort of host immune defenses, right? A virulence factor is really a generic term for anything structurally or chemically a cell or a pathogen does to help be dangerous, I guess is a good way to say it. Now, exoenzymes can also be toxins, right? So those can be some type of chemical that gets pumped out and might bind to neurological receptors, right, and inhibit something inside of your body and, can cause damage that way. Now, enzymes are sensitive to their environment, right? And so that's why it's actually pretty difficult to figure out the structure of a protein in an enzyme, because as soon as you pull it out of the cell, you put it in different conditions, it's going to change its shape and what it looks like. How we actually look at proteins and look at enzymes is by using something called X-ray crystallography, is the most common method, where we essentially crystallize it before we pull it out and look at it, because temperature, pH, osmotic variations, all those things we kind of talked about in the microbial nutrition part of the course are things that can impact the function of an enzyme. And since enzymes and proteins are so important to the function of a cell, right, it's really detrimental, especially to these unicellular microbial organisms, to move sort of outside of their range, right? Then their enzymes stop functioning, their proteins stop functioning, and it can be potentially dangerous. Now, when they move sort of outside and become a little bit unstable, it's known as being labile, but even beyond that, they can then denature and they change their shape and they can no longer bond to their substrate. All right, so within the cell, these proteins and enzymes are what assist in all of these chemical reactions within a cell. So a metabolic pathway is a pathway in which we, perform jobs within the cell. You think about it like a factory line rather than like each enzyme having, one function, or I guess achieving something overall, right? We need step-by-step process because it is so specific what those enzymes do. So typically to get from point A to, point B in the cell, you're going to have a lot of different enzymes that have one little role. So if you look over here in the linear pathway, it's like A, B, C, D, E. Essentially, each one of those letters is representing one enzyme that performs one small little step in an overall, metabolic reaction or pathway. We have cyclical pathways, right? You've probably, like the Krebs cycle and glycolysis, we're going to talk about. But each one of these indicates a different enzyme where at a different step performs one very specific function. We also have branched metabolic pathways, so where you have maybe starting on a linear pathway, but then whatever enzyme that is, it can lead into different, products, or some that converge, so divergent and convergent. So there's all sorts of different pathways in order to achieve different goals within the cell. Now, when we develop drugs for bacteria, a lot of what we're trying to do is we're trying to inhibit some of these pathways, in really essential pathways that are going to do damage to, that microbe. So we can design drugs that will inhibit something inside of that pathway. If you cut off, like in this linear pathway, if you cut off the function of enzyme at position C, right, then, this pathway would be shut down. Okay, so, that's what we try to do, and not necessarily specifically, like we don't always know what the drugs are going to do or what enzyme they're going to bind to. That's actually pretty rare. That we can do that, but we throw a drug at it, it shuts down a pathway, and we're like, okay, this happens, right? This drug works on something in there, okay? So it's a little bit more blind than I think some people think, but we'll talk more about this when we do like drug development, right? But those are the different types of pathways that we're looking at. So we have ways of inhibiting enzymes by either competitive or non-competitive inhibition. So competitive inhibition is when you actually have a compound or something goes in and it blocks the active site of the enzyme, right? So the substrate can't even get into the enzyme. More common we have is non-competitive inhibition. Non-competitive inhibition is when something binds somewhere else on the enzyme, and since their structure is so dependent on, what they're sort of interacting with, it's very specific. As soon as you attach something to the side of it, it's going to change its shape, and it essentially makes it so that the active site is no longer the correct site to bind to the substrate. So if we talk about this in terms of drugs, right, if I design a drug and I want to block a pathway, I will expose a cell maybe to that drug, and that drug binds somewhere, most likely somewhere non-competitively. Like we don't know exactly what the shape of the active site is, right? We're not specifically designed drugs that are going to look exactly like that substrate and be able to bind in there. We're just throwing chemicals at enzymes, and if those enzymes, if they attach to the enzymes, they shut that enzyme down, right? And there's this non-competitive inhibition. So that's why I was saying it's more of like a blind approach, right? We try a whole library of different chemicals to see if they'll just stop it, right? However, there are some where we have what's called a metabolic analog, where we have studied the enzymes so in detail, or the pathways so in detail, that we've been able to actually design a drug that, competes for the active site. Okay, so that's looking at this in terms of drug development. Let's talk about it in terms of regulation within the cell, right? So sometimes we want enzymes functioning, sometimes we don't, right, within our cells. So you can also regulate those enzymes, like I said, upregulate or downregulate, by having regulatory molecules that would bind somewhere else on the enzyme, and essentially, changing the shape of the active site and making it, either turn on or turn off, right? So we can think about it in terms of a function within the cell, but also as in terms of drug development when we're trying to shut down pathways in potentially hazardous pathogens. All right, so if we look at the repression and induction, to give you kind of an idea of how this works, there's a couple of different ways in which this can happen. We can bind something to the side of an enzyme or and regulate it like this, turn it on or turn it off. But we can also, increase the formation of or decrease the formation of the enzyme. And this is essentially going to be some sort of interaction between the product or substrate and the DNA, okay? So the DNA carries the message that is then going to be, carries the information that's then going to be translated into a message that is transcribed into a message and then translated into a protein. Sorry, I just said that horribly. Let me say it again. The DNA has the information that then is transcribed into a message and translated into a protein. So the presence of a product could bind to the DNA in the location of the gene where we make that enzyme and turn it off. Say, don't work, gene. I don't need you around. There's enough product here available. I don't need this enzyme right now. The other thing that it could do is there could be a substrate present, and it could promote the transcription of the DNA into a message that then would make the protein, right? So that enzyme induction is when we have, trying to force that enzyme to be made by interacting with the DNA, whereas the enzyme repression is the presence of product being present and shutting down the gene that it would then, send the message to make that specific protein or enzyme. All right, in the next video, I'll give you a little bit of an introduction to metabolism, and then we'll get into the electron transport chain and other ways that ATP is formed.